Bacteriological characteristics of water. As per the data of WHO 2019, 2.2 billion people are not having access to safe drinking water. Therefore, treating the available water in an efficient way is the need of the hour. The water quality should be tested in terms of physical, chemical, and microbiological aspects. But among the three, microbiological aspects should be given more emphasis as it have direct impact on human health. The biological characteristics of water refer to a variety of living organisms that can be found in water. These may include microscopic viruses, bacteria and protozoan, zooplankton, insects, worms, plants, fishes, etc. The microbiological examination of water. It simply means the determination of presence of bacteria and other microorganisms in the water, and it includes bacteriological determination and biological determination. Then comes the classification of bacteria. Based on the shape, based on the nature of bacteria, and based on the supply of oxygen, the classification of bacteria is done. First one, based on shape. The few different kinds based on the shape are cocci, bacillus, spirilla, and based on the nature they are classified into two, harmless bacteria and harmful bacteria. Based on the supply of oxygen they are classified into three, aerobic bacteria, anaerobic bacteria and facultative bacteria. First one, based on shape, cocci. These are the spherical or oval shaped bacteria, and these bacteria cause diseases like strep throat, pneumonia, meningitis, food poisoning, and they can also even cause skin diseases and septic shock. Bacillus. Bacillus are rod shaped bacteria, and these are the major cause of diseases like diarrhea, abdominal cramp, vomiting, nausea. Then comes these bacteria causes cholera, tuberculosis, strep throat, staph infections, food poisoning, etc. And the examples of these bacteria are Escherichia coli, or simply called as E. coli, hepatitis A, Campylobacter jejuni, Salmonella, Cryptosporidium. Then comes harmless bacteria. Harmless bacteria are non-pathogenic bacteria, that means they do not cause any kind of diseases. Example, Staphylococcus epidermis. Based on the supply of oxygen, aerobic bacteria, anaerobic bacteria and facultative bacteria are the three divisions. The aerobic bacteria are the bacteria that grows only in the presence of oxygen, and the end products of these bacteria are carbonate and nitrates, and these end products are very stable while decomposing. Second one is anaerobic bacteria. These grows in the absence of oxygen, and the end products are like carbon dioxide, H2S, methane, etc., which are unstable during decomposition. Facultative bacteria. Facultative bacteria can grow under both aerobic and anaerobic condition, and these are the bacteria that use dissolved oxygen or oxygen from food materials. Waterborne diseases. Waterborne diseases are caused due to the bacteria from contaminated water supply. Waterborne infections account for 2 to 3 billion episodes of diarrhea, resulting in 1 to 2 billion deaths a year. Contaminated surface water source and large poorly functioning municipal water distribution systems contribute to transmission of waterborne bacterial diseases. Chlorination and safe water handling can eliminate the risk of waterborne diseases, and the microorganisms usually present in this contaminated water include protozoa, bacteria, and many viruses. And these are intestinal parasites or invade the tissues of or circulatory system through the walls of digestive tract and cause serious illness. The bacterial infections are typhoid fever, cholera, diarrhea, bacterial dysentery. Viral infections are hepatitis, poliomyelitis, etc. The protozoal infections are amoebic dysentery. The major diseases caused by the pathogenic agent and their symptoms and prevention. Typhoid fever and its pathogenic agent is Salmonella typhi. Symptoms are fever, muscle ache, fatigue, sweating, diarrhea or constipation. And this can be prevented by vaccination, avoid eating open food and drink clean water. The cholera and this disease is spread through contaminated water and cause severe dehydration and diarrhea. It is mainly found in humanitarian emergencies or marginalized villages where poverty or poor sanitation are ramped. And these are caused by Vibrio cholera and the symptoms are nausea, vomiting, diarrhea, and this can be prevented by washing your hands properly, eating food that are completely cooked, drinking safe and clean water, etc. Dysentery. The main these diseases are spread through contaminated water and poor hygiene. It can be caused by bacteria or virus or by people coming in contact with fecal matter, and it is an intestinal infection characterized by severe diarrhea as well as blood or mucus in the stool. And this is caused by Shigella dysentery, and the symptoms of this disease are stomach cramp and pain, diarrhea, fever, vomiting, dehydration, nausea. And this can be prevented by washing your hands often, don't eat foods sold by vendors, drink clean water, etc. Then comes E. coli. E. coli is a bacteria with various strains, some dangerous and some beneficial. For example, E. coli is important in creating a healthy intestinal tract. The bacteria is found in unsafe water sources around the globe where human water source and cattle coexist. And these are caused by the pathogenic agent Escherichia coli or simply called as E. coli. These are similar to dysentery in symptoms. The prevention methods are avoiding the water possibly contaminated by human or animal feces, washing fruits and vegetables properly. Salmonellosis. These are caused by salmonella and these are caused Salmonellosis. Salmonellosis are mainly caused by ingestion of food or water contaminated with fecal matter. Children, pregnant women, old adults are the ones who are mainly affected by this disease. And the major cause of this disease are in contact with birds, contaminated water by animal and other human feces. The pathogenic agent is Salmonella and the symptoms of this disease is blood in stool, headache, diarrhea. Detection of chloroform bacteria. The presence of bacteria in drinking water is harmful to human health. Escherichia coli is the name of a bacterium that lives in the digestive tracts of humans and animals. And these have the strains that are useful as well as harmful. And these harmful strains should be detected. Otherwise, it can cause severe anemia, kidney failure, and even other diseases, and that can even lead to death. So it is important to find the presence of E. coli in drinking water. The methods used for the detection of chloroform bacteria are membrane filtration and multiple tube fermentation. Membrane filtration method. It can be defined as the process of microbiological analysis of water by making the use of a special filter like membrane filter to trap organisms. In this method, we use a membrane filter and it is made of cellulose. So it have fine pores so it can retain the microorganisms. Water sample is passed through sterile membrane on which bacteria will be retained. The filter is then put in nutrient solution. Nutrient solution for total coliform as M-ento broth and nutrient solution for fecal coliform as MFC broth. Then it is incubated at suitable conditions and visible colonies are counted with microscopes. The simple methodology can be simply explained as: first we have a flask, we should have a funnel, then assemble the filter funnel on the flask, then place the sterile membrane over the carbon disc. Then the water sample is allowed to pass through the funnel, filter under the gentle vacuum. After filtration, remove the membrane and then it is incubated and cultured in a prepared appropriate medium. After incubation, count the colonies to determine the concentration of organisms in the same. The second method is multiple tube fermentation method. It is also called as most probable method. And in this method, it enumerates coliforms, especially E. coli. The water to be tested is diluted serially and inoculated in lactose broth. The coliforms, if present in water, utilize these lactose present in the medium to produce acid and gas. The presence of acid is indicated by color change of the medium and the presence of gas is detected as gas bubbles collected by the inverted tube present in the medium. The number of total coliforms is determined by cutting the number of tubes giving the positive reaction and comparing the pattern of positive results. The NPN test is performed in 3 steps: presumptive test, confirmatory test and fecal chloroform presence test. The presumptive test, the broth containing lactose and nutrient medium is placed in a series of tubes, that is 5 tubes with 10 ml samples and 5 tubes with 1 ml sample water and 5 tubes with 0.1 ml water sample. These tubes are incubated for 24 hours and formation of gases are noted. Then the presence of gas will give an indication of presence of coliform. If the gas is not formed, it again incubated for another 24. If no gas is evolved, it will show a negative test indicating the absence of coliform. Then comes the confirmatory test. A portion from the tube showing positive presumptive test is mixed with brilliant green bile broth and incubated for 48 hours. Coliforms are gram-negative bacilli which have the ability to ferment lactose with the production of acid and gas. If gas is evolved, it will be positive confirmatory test confirming the presence of chloroform, and if not, it will be negative test. After confirmation, the density of chloroform is determined and it is represented as MPN or most probable number, and through which we can find the presence of coliform. Then comes fecal coliform presence test. A portion of the tube showing positive presumptive test is mixed with easy medium broth or Escherichia coli broth and incubated for 24 hours. If gas is involved, it will be positive confirmatory test confirming presence of fecal coliform, and if not, it will be negative. Thus, these two methods are very effective method for isolation and enumeration of microorganisms from the test water samples. Maximum acceptable concentration for drinking water is that for every 100 ml of drinking water tested, no coliforms or E. coli should be tested. And thank you. Thank you.